Hi class. So we're on the topic of enzymes, and normally we would do notes together in class, but I think I'm just going to go through these notes with you, and you'll just sit back and watch them, and these will be a resource for you. So let's talk about these enzymes here. I'm going to remove my picture, I think, so that I'm not in your way. Hide this out of the way. Okay, so enzymes we're considering them helper protein molecules. They help chemical reactions occur, and they speed everything up in our cells. Without these enzymes, nothing would happen in our cells, and we wouldn't be able to survive. So the flow of energy through life is built on chemical reactions, and for everything to function, like I said, is built on chemical reactions. So when your cells are building molecules, we call that synthesis. And I'm going to get my little laser pointer here. Synthesis is going to be an important term. You need to remember that. So if your cell is synthesizing something, it's making something. And when you're breaking things down, you're digesting them. That's breaking them apart. So you can see that in these pictures. The first one is showing you these two shapes are separate from each other, and when your cells synthesize something, they put them together with a bond and you create something bigger. And then digestion is taking this larger molecule and breaking it down into two smaller ones. All right, so nothing, like I said before, nothing works without enzymes. All chemical reactions in our living cells require enzymes to work. So we have enzymes that help synthesize. There are different enzymes that help break things down and digest. Enzymes that speed up reactions in a living thing are called catalysts. And that's going to be an important term down here. The catalysts are, you're going to want to remember that term. Sometimes they're called biological catalysts. So here's some examples again. The first picture is showing you an enzyme helping these two molecules join together. This is synthesis. And this is showing an enzyme helping to break these molecules apart in digestion. Each enzyme is a specific helper to a specific reaction. Each enzyme has to have the right shape for their job. They have to be very specific. Enzymes are named usually for the reactions they help, which is good. So, for example, sucrase breaks down the sugar called sucrose. And I just want to add in, I know your Amoeba Sisters video tells you this, but anything that ends with ASE is typically an enzyme. So you're going to want to make note of that somewhere in your brain or on paper. And anything that ends in OSE tends to be a sugar when we're looking at science words. So like glucose, sucrose, fructose, those are all different types of sugars. Okay, and then we have a protease enzyme. Notice it has an ASE. It'll break down proteins. We have lipases that break down lipids or fats. And DNA polymerase actually builds DNA, polymerase, so it's going to make a bigger molecule. And it's interesting too, because if you look at this reaction, the enzyme is not used up and it doesn't really get changed by the reaction. So it can be used over and over again. And they're used temporarily. Like I said, they can be reused. And it doesn't take a lot of enzyme to help lots of reactions because they can be recycled. And we're seeing here this cutout area is being labeled as the active site. So that's something to make note of. That little cutout area where the substrate can fit into perfectly is called the active site. The yellow thing or the white thing is called the substrate. That's the thing that's our reactant. That's what's going to be taken apart or put together, depends on what reaction we're looking at. The enzyme is the purple thing, and then our product is what we get at the end. So it is the shape that matters. If you think about a lock and key model, only one type of key can fit in one type of lock. and you have to have the right lock or the right key to open the right lock. So just like with proteins and enzymes, the shape is going to be very important. You have to have the right shape for it to do its job. So there are specific enzymes for each specific reaction that occurs. In this diagram, we'll take a look here. Our substrate is sucrose, which is a type of sugar, and it fits into this enzyme. It's called the enzyme substrate complex when they're all bound together. And then through another chemical reaction, the enzyme's starting to pull those two sugars apart, and you end up with glucose and fructose at the end of this reaction. And then you can see the enzyme gets recycled and ready to go around again. Some vocabulary terms that you want to make sure you remember. You could always make flashcards out of these. We have enzymes. Those are helper protein molecules. The substrate is the molecule that the enzyme works on, or you could also write reactant. The products are going to be what the enzyme helps produce from the reaction. And the active site is the part of the enzyme that substrate molecules fit into. Okay, there are things that will affect how the enzyme works. You have to have the right protein structure, because if you don't have the right structure, it's not going to have the right shape. And remember, proteins are made of amino acids, and enzymes are a type of protein, so they are made of amino acids. If you don't have the right amino acids put together, you're not going to have the right shape for its function. So, like I said, you have to have the correct order of amino acids. And as I said before, it has to be the right shape. Temperature. Temperature and pH can affect the shape of the enzyme. If something is too cold, that enzyme is not going to work very well. There's not enough energy for the reaction to occur. So it's going to occur very slowly or maybe not at all. But if you increase the temperature to a point that is above the optimum or above the best temperature, then those proteins are actually going to start to denature. They're going to fall apart and change their shape, and then they're not going to work at all, and they can't be changed back again. Think about an egg. Eggs are mostly protein, and if you cook an egg, they change. Their protein structure changes, and you cannot change it back. So that's why temperature is very important. And pH, the same thing with pH. If it's not the right pH, it can denature the enzyme and change its shape, and then it won't work either. So these three things are going to be important to remember, that the shape of the protein or the right amino acid structure is going to be important. And then the temperature and the pH can affect how an enzyme functions. This diagram is just showing us we need to have the right order of amino acids. And the instructions come from DNA. The DNA are the instructions of how to put a protein together, what is the order of amino acids, and then based on that structure, the protein can fold up into its correct shape. If there's a mistake in the DNA, it's going to fold differently, and then it won't have the right structure. But we have thousands of different types of proteins, so different parts of the DNA are going to have the code for different types of proteins. So like I said, if the amino acids are in the wrong order, you're going to have the wrong shaped protein or amino acid or enzyme, and then it can't do its job. So for temperature, they're going to affect the rate of enzyme activity. There's always an optimum temperature. That's the best temperature. And this is when you're going to have the greatest number of collisions between the enzyme and the substrate. For example, human enzymes work the best at our body temperature, which is somewhere between 35 and 40 degrees Celsius. Our main body temperature, the norm, is 37 degrees, or if you convert it to Fahrenheit, that's 98.6 degrees. And so if you think about it, our enzymes are happiest at that temperature. But if you get a fever and you start to feel very sick, it's because some of your proteins are not functioning properly, and they could end up denaturing if your temperature goes too high. So think about raising a temperature really high to like boiling. You're totally going to denature that protein. It'll completely lose its shape and it won't work. If you lower the temperature too much, the molecules move very slowly, and then you don't have very many collisions between the enzyme and the substrate, and then you're not going to have any reactions. So this is showing us our optimum temperature at 37, and the reaction rate is going to be pretty slow if it's too low, and it's going to be pretty low if it goes too high because the proteins denature. Think about cold-blooded creatures like snakes and other reptiles. They have to acclimate their body to their surrounding temperature. So they have to be warm enough for reactions to occur in their cells. Otherwise, those reactions don't occur and they could end up dying. That bobcat is showing what warm-blooded animals can do. They maintain their body temperature at a regular temperature. It doesn't matter what the outside temperature is. So their cells can always function at that particular temperature, and the enzymes function best at that 37 degrees temperature. Okay, so pH is going to affect the shape of your enzymes and how they function. So a change in pH will change the protein shape. And in our bodies, most human enzymes are going to work best between a pH of 6 and 8. Not too acidic, not too basic. Except certain areas, like in your stomach. Pepsin, which is a digestive enzyme, works really well at a pH of 3, which is very acidic. And trypsin, which you can find in your small intestine, it helps break down some of the proteins. It's going to work better at a slightly higher pH, slightly basic. So there's a range in our bodies, but there are certain areas where that middle ground is when the enzymes work the best. So here our optimum pH would be neutral, and then for most cells, most areas, they're gonna function the best at that center area and decrease either if it's too acidic or too basic. So this is showing us pepsin and trypsin and how they vary from that center area. Pepsin is going to work the best at a very low pH because it exists in the stomach where it's very acidic. And trypsin is going to be found in those small intestines where it's a little more basic. There's not so much acid there. And it functions at a pH of 8 at the best. So for enzymes, what matters? It's the shape that matters. It's very important.